Okay, and welcome to another Biome webinar series. So really happy to have all of you. For those of you who, welcome May as well. Nice, people are rolling in. Okay, so my name is Alan, and I'm a lab manager at Biome. And today we're going to be talking about another lab technique. All right. Now, this is what you all came for, right? You all came to learn how to be a DNA ninja, right? And so for those of you who are interested, I'm going to post a link in the chat. If you aren't already on our emailing list, then that's where you can fill out the form. All right. So let's get started. I'm going to keep the chat on the side. So if you have any questions at all, please ask them in the chat, and I'll try to keep an eye on that. And also, if you wanted to speak out and tell me, that's also an option too. Okay. So what are the steps to becoming a DNA ninja? Well, first we have to understand some basic biology. And so I will remind you guys about DNA. And so we all know that we are DNA, right? DNA is sitting in the nucleus of our cells. And we can read DNA like a book, right? DNA has letters, just like a book has letters. But those letters are A, C, T, and G. But those aren't just letters, they're molecules, right? They're atoms bonded together in certain ways. So we can take a look at this structure here. This is a picture of the molecular representation of DNA. And so I can annotate on the screen. This right here, I believe is an A, and this right here is a T. Here is a G, and here is a C. So we say that DNA is complementary because A always pairs with T and G always pairs with C, right? It's complementary. They have their own buddies to cling on to, right? You rarely see A binding with, C or G. So my question to you guys is why? Looking at these structures, what's holding these two together? Anyone know what those are? You can text in the chat or just unmute yourself. Both are okay.
Hydrogen bonds.
Yeah, perfect. Thanks so much. And so these hydrogen bonds are holding those two strands of the DNA together, and we all know that it's the double helix, right? So that forms this really iconic structure. Any questions about DNA? I think we all have heard about this before. That was just a really quick review. But now I want to go on kind of a tangent, right? I want to talk about your immune system. And so our immune system, very relevant, right? Because of COVID-19. The reason why we're all in quarantine is because we don't have strong enough immune systems to fight off COVID-19. And so our immune system, it has a really, really important responsibility. And that is to sense, is this me or is this not me? Right? That's like the Descartes question for your immune system, right? To be or not to be, not the immune system. They ask, is it me or is it not me? So if your immune cells detect that it's you, then it will not attack it. Simple as that, right? If it's not you, it will attack it. It will assume that it's what's called a pathogen. So I think we all have heard about the immune system before. But my question is, do bacteria have immune systems? How about you guys press participants? You should be able to say yes or no. Let's see how many say yes, how many say no. Do bacteria have immune systems? I'm seeing one yes. Maybe take a few seconds to think. Okay, 2 yeses, 3 yeses, 4 yeses. Oh, nice. 5 yeses. Okay, an overwhelming number of yeses. And you guys are correct, right? It's kind of weird to think about because we typically think about organ systems as multiple cells, right? But a bacterium is just a single cell. And so it's kind of weird to think about how they might have immune systems, but they do. It turns out that it's molecular. It's not cellular. And so here we have our happy bacterium, bacterium singular. He's going about his day. He's very happy. But then he gets sick, right? Does anyone know what kind of things can make bacteria sick? What are they called?
Bacteria.
Right. Yeah. So Alex said bacteriophages, and you said viruses. Exactly. Bacteriophages are a type of virus, and they look like this, right? And so these alien-looking drones go down on the bacteria, and they infect the bacteria. They go into the bacteria, and they hijack their system. And the bacteria now isn't feeling so good, right? The bacteria is now like, oh my gosh, I have all these viruses inside of me. And then viruses are wild things. They can replicate inside of the bacteria until the bacteria just blows up, it lyses. and then more, viral progeny just come out. So the question is, what's the immune system of the bacteria? How do they prevent this from happening to them? Right? Because viruses are everywhere. Believe it or not, viruses, they are everywhere. So this is what they do. Okay. Now this might seem like a tangent. Like, I want to know how to be a DNA ninja, right? What am I doing talking about bacterial immune systems? This will show you the power of basic biology, right? So here's what they do. They use a protein. It's an enzyme, and they call it a restriction enzyme. You'll see why soon. So the restriction enzyme is the bacterial equivalent of a DNA ninja, right? This is how they do it. They express this protein that can bind to DNA, and then they can use their ninja skills, and that's their immune system. Let me show you what I mean, okay? So this is their weapon, this protein here. Is it about CRISPR? Yeah, you're reading my mind. So that will be later on the talk. We'll first talk about restriction enzymes, then we'll talk about CRISPR, which is really it's almost like a subtype of restriction enzymes. So, good point. This is what happens. Okay, so first, your enzyme, your protein is floating around in the bacterial cell, and it finds the DNA. And it searches for that DNA, and it finds that DNA, and then what? Then it activates ninja mode, right? Boom, boom. And then ninja mode activates, it slices through that DNA, and then that DNA is broken in half, right? So this restriction enzyme has the ninja power to slice that DNA strand in half. And so we call it an endonuclease because it cuts, it's an enzyme, so it catalyzes some reaction. It cuts inside of the strand, it's somewhere inside, right? It's called an endonuclease. An exonuclease will be cutting from either end, right? So that's what the difference between endonuclease and exonuclease are. For the most part, we're going to be using endonucleases, but exonucleases are also really important. Okay. So these restriction enzymes are the bacteria DNA ninjas, right? They're going around chopping up the DNA, right? It turns out that they can recognize very specific sequences. And when that sequence is inside of a virus DNA, they will recognize it and cut it, right? But my question is, I almost spoiled it for a bit. My question to you guys is, why doesn't the restriction enzyme cut the bacteria's own DNA? Because it recognizes a site that's about maybe 5 or 6-ish base pairs, right? 5 or 6-ish A, C, T's, and G's. I mean, the bacterial genome is a lot bigger than that. There's bound to be some of that in the bacterial genome. How does the bacteria not attack itself? Anyone know? Again, this gets back to the idea of immune systems, right? What is the defining feature of immune systems? It's that we have to differentiate self versus non-self. For the bacteria, self is the bacterial DNA and non-self is the viral DNA. So the key is the bacteria must have some mechanism to tag their own DNA, right? And what they do is they methylate it. It turns out that this methylation, they add methyl groups, these blue blobs, they add these methyl groups on their own DNA to stop their own restriction enzymes from cutting it up. So here's a more nuanced picture. So the bacteriophage injects its DNA, and then your ninjas go to work by chopping it up, right, into these fragments, which are then further degraded. But your bacterial DNA is protected because of those methyl groups. They actually prevent that enzyme from binding. That's how it works. Okay. Now here's an example of what a restriction enzyme might see. So this EcoR1 is a type of restriction enzyme. Why is it called that? Does that seem familiar to any one of you? EcoR1. It might sound familiar for those of you who might have read some biology before. EcoR1. Anyone? E. coli, right? Alex, is that what you're about to say? Yeah, yeah. E. coli. So this was a enzyme that was taken from E. coli. So we call it eco, and then R1, because it was some strain and then some number, right? So this E. coli restriction enzyme, it recognizes a specific sequence. Specifically, it's GAATTC, and it cuts at that point. It uses its DNA ninja skills to cut at that point. Now, Andrew asked a very good question. Why don't viruses develop methyl groups on their DNA? It's a very good question. I think that they don't have the machinery to do so, right? Because viruses are pretty simple. They only have a few enzymes, a few proteins. So I doubt they have the machinery and the cellular work power to put methyl groups on their DNA. Although I think you're on a very good point. Right? So Andrew, I like your train of thought, because viruses, they have their own genetic code, so they can mutate. So if you think about it in that sense, it's almost like an evolutionary arms race between viruses and bacteria, right? Because the bacteria developed, they evolved this restriction enzyme system to counteract the viruses. But as Andrew mentioned, well, what do the viruses do, right? Do they just die? No, the viruses have to also have some kind of counterattack, right? And so, Andrew, I would expect that there was some other kind of mechanism to disable these restriction enzymes, and those do exist. And you also mentioned CRISPR before. Right? So bacteria, we'll get into this more later on in the talk, but bacteria also have this other type of immunity called CRISPR. And guess what viruses evolved? Anti-CRISPR. So it's an evolutionary arms race, right? So these different lineages, they're keeping up with each other. It's not like, you know, the bacteria win and the viruses all die out, right? That's not how it works. They're evolving alongside each other, trying to infect and trying to, you know, survive. So it's a harsh world out there. Actually, you know, one thing related to that is the idea of phage therapy. Have you guys heard about antibiotic resistance before? Maybe can someone really briefly describe antibiotic resistance for those who might not have heard about it before? You can text it or say it. And this might take a while to type up, so I'll give it a few seconds. What is antibiotic resistance? Antibiotics kill bacteria, right? Antibiotic resistance. So Alex says, organism blocks their own receptors so that antibodies can't bind to them. I wouldn't say it's that. I would say antibiotic resistance is more of a evolutionary mechanism. So pretend you have some bacteria and you add some antibiotics, the weaker ones die off and the stronger ones live, right? Survival of the fittest. And then eventually, over many, many generations, as they divide and divide and they mutate even more, they'll become more and more resistant to your antibiotics. So Andrew says bacteria reproduce really quickly, so some mutate and survive to antibiotics. That's a huge problem, right? If you have some bacterial infection, you want to be treated by using antibiotics. But if those bacteria are these so-called superbugs and they're resistant to your drug, then you can't really cure yourself of that illness, right? That's a very huge problem. And so I guess, Andrew, going back to your old point, right? People have exploited this before. They think to themselves, well, these bacteria are evolving against our drugs. Well, what evolved against bacteria? Bacteriophages, right? So there's the idea of phage therapy, where these scientists design these bacteriophages to specifically target those bacteria that are, you know, antibiotic resistant, and that might be able to save a lot of lives in the future if that happens to be the case. So I feel like thinking along those lines of, well, how do we take advantage of these natural systems? That's the whole point of synthetic biology, right? All right, awesome. Sorry for that tangent there. Good point though. So let's keep going. So it recognizes this sequence, GAATTC, and then CTTAG on the bottom strand. What's fishy? You guys see anything? What's fishy about that sequence?
It goes all the way to.
Yeah, right? Check this out. We have here GAATTC. If we read it backwards, it's GAATTC, right? It's a reverse palindrome. It's not an exact palindrome, it's a reverse palindrome. And so you'll oftentimes see these in terms of restriction enzyme sites. You'll see these reverse palindromes. Sometimes you'll have regular, as we call them, palindromes, but oftentimes you'll also see these reverse palindromes. And you can see how eco R1, it cuts at this spot, right? And so you form these DNA fragments. What's important about these DNA fragments? They can do what? Why does that overhang make it really important? Right? It's GC, but then you have this like bit of extra shoelace coming out, right? Why is that extra bit of shoelace important? Yes, May.
It makes it like a sticky end. So if you want to insert like a gene, as long as it has the complementary ends, it can stick.
Yeah, exactly. We call them sticky ends because they're sticky, right? They can hydrogen bond to each other. Thank you, May. Perfect. So here is a more sort of chemical lookout of how this works. So you have your sequence of DNA, right? This is what, A, G, C, T, right? And then you have some endonuclease, some restriction enzyme. And the mechanism actually is not too important for our discussion here. It is really fascinating. But they actually take a water molecule and they basically stuff it inside. It's a hydrolysis reaction. They stuff it inside and that breaks that bond. So some people also might call these restriction enzymes as a larger class phosphodiesterase because they cleave that phosphodiesterase bond. And so you're left with these hydroxyl groups and these phosphate groups at the ends of your strands. For those of you who have taken biology before, do you recall what we call these ends of the DNA? It's a numbering system, right? What would we call this end?
Is that the 5' end?
It is. Yeah, perfect. So we count starting from here, right? 1, 2, 3, 4, 5. So this is what we call the 5' end of the DNA. And that makes this side the 3' end, right? Here is the 1, 2, 3. So we call that the 3' end. And this is the 5' end going away, right? Awesome. That's just some nomenclature, so you'll get used to it in the later slides. So anyways, the overall restriction enzyme mechanism is a hydrolysis reaction that cleaves that phosphodiester bond. Okay. Of course, this is only showing one strand, right? This is a double strand of DNA, so this is only showing one, but on the other side it's also happening. There's also that cleavage of the phosphodiester bond. Okay, let's move on. Here's just a table. You can just Google these up. They have entire lists of these online that you can choose from. So you can see how some of them don't make sticky ends, they make blunt ends, and that's actually kind of annoying because you don't get that sticky effect. They are useful in some situations, but you can see how most of them are sticky ends, right? You can see how most of them cut away from each other. Here's a blunt end one again. And they all have their own names. You can order them online if you're doing some research. But again, that kind of gets me back to the question, how do we use this? And May kind of touched on this earlier, right? It's the idea of genetic engineering. So of course, back to our discussion about our sad bacterial friend, now the virus is dead. After you throw all those DNA ninjas at it, now the virus is dead, which is good for the bacteria, right? The bacteria is happy again. And I guess viruses aren't living, so the viruses don't have feelings. So we're all good. How do we use this? So May, you kind of foreshadowed it, right? We have the idea that we can use these restriction enzymes to insert genes. You guys have seen plasmids before, right? These circular pieces of DNA. Every single one of these sites is a restriction enzyme site. If you go to any plasmid map and you zoom in, it will show you exactly where those sites cut. You can see how useful these will be, right? You can see, oh, if I want to insert a gene here, I can simply use those restriction enzymes. Question. If I use a restriction enzyme on a circular plasmid, what will my end result be?
A strand.
Exactly, right? You've linearized it. So you have a circle in the beginning, you nick it off, you cut it, now it's a line, right? Now it's a linear strand. And so, oh, there's a good friend, eco R1, right? And so we can see how all these restriction enzymes have specific sites along the plasmid. So say you want to insert one right here, right next to the promoter, you can do that by looking at what sites are nearby. Okay. Now here's the idea. Here's the one that May alluded to earlier. We have some DNA that we want. This is our gene of interest. We do PCR. I won't go into PCR in this lecture. PCR we might go into later on, but PCR is a way of amplifying DNA. You make a whole bunch of it, and then you digest using your restriction enzyme. That's sort of another word you might encounter, digestion. You just cut it up. You use your DNA katana, right? And that chops off this point. And you use the same, and here's the important part, right? You have to use the same restriction enzymes on both your gene of interest and your plasmid. So then you form the same sticky ends. You see how that works? Then you mix them together, and then there's this process called ligation, where you then just combine them together. So you see how this sticky end will be complementary to this sticky end. And this sticky end, I'll do a different color. Right? This sticky end will be complementary to this sticky end. That's how they will bind together. And once they hydrogen bond, then you reform that phosphodiester bond using ligation, sticking together, right? And then you have what's called a recombinant plasmid, which you then can give it to some bacteria for them to express that DNA, right? And on this side, it's showing the same thing, but on a more close-up scale. You have these molecular scissors. People call these restriction enzymes molecular scissors. Right? You can chop through a very precise sequence, and then you can ligate it to your plasmid after chopping the plasmid up with those same scissors. And then you have your complete plasmid, right? Okay. So this is genetic engineering, right? We are inserting whatever gene. Think about how powerful this is, right? Choose any gene you want. You can put it into a plasmid. You can put it into a bacteria. That bacteria can then start doing your bidding, right? Okay. And here's just another close-up. Right, here we have, we cleave it with our EcoR1 restriction enzyme. As we know, that cleaves GAATTC. That cleaves it like this. Right? On the other side, we also cleave it like that again, this way, right? So this is our gene of interest, say for example, and then those can hydrogen bond, and then we can form that phosphodiester bond between the bases, right? And so that's caused by DNA ligase. So that's another enzyme you have to add to the mix. The ligase forms those bonds, so those two don't come back off again. Right? They're now stuck together, and so then you form your complete plasmid. Okay, so digestion, ligation. Do you guys see a problem with this model here? What's an issue you might encounter? Right? You cut up your DNA, you cut up this plasmid DNA, and let's say, for instance, that you cut using okay, anyways, regardless, assume you're using the same enzyme, right? You assume you're only using one restriction enzyme. What problems might you encounter if all of this is in the same mix? So you cut this DNA, you get those sticky ends. You cut this DNA, you get those sticky ends. What can happen? Yes, May.
Some of the plasmids might just fuse together and not get the gene in.
Right, exactly. If this vector here recircularized, that's not good, right? Because we want our gene inside of that place. We don't want it to recircularize. So there are ways to get around that, and I'll get to that in a bit. But for now, here's again the mechanism. So you can see how your ligase, right? You want to fuse them together. How do we fuse it? Your ligase has an amino acid called lysine in it somewhere. Your lysine attracts this ATP molecule. Right? You all heard about ATP before. That's the energy currency of the cell. And it uses this ATP to then catalyze this reaction. It does some phosphate transfer reactions, and then the leaving group is, I believe it's AMP. So then you form your phosphodiester bond between those two strands. So this is your ligation event, right? So your ligase has to find where there's that break, and it also has to detect where the phosphate group is, brings them together to form that ligation event, right? Enzymology, there's a whole lot more you can go into, but this is sort of a scheme that shows how it works overall, right? So again, we go back to the question, how do I prevent the plasmid from simply going back together, right? That's not good. We want our gene inside, but sometimes, I mean, if you think about it, it's more likely for them to recircularize, right? Because it's right there. It's an intramolecular attack, which is more likely than an intermolecular attack. So how do I do that? Any thoughts? Look at the mechanism here. Right, this is how ligase works. Ligase, it looks for a phosphate group to attack, and then that develops that phosphodiester bond. What might be a way to prevent recircularization?
Could you break the ligase protein so that it can't bind there?
You could, yeah, but if you break the ligase protein, then you can't ligate in the end either, right? So we still want ligase to be in there. We still want the ligase to be functional. But what else can we change? So we can't change ligase. What can we change? How do we prevent recircularization? I'll give you all a hint. The ligase recognizes the phosphate group, right? That's what it's using to bind them together. Any ideas? Andrew says change the phosphate on the circular DNA. You're exactly right. Let's just chop it off, right? Let's use a phosphatase to chop off those terminal phosphates. And that leaves you with these hydroxyl groups at the end, right? So you can see how if we don't use the phosphatase, sometimes it will just simply, through ligase, it will simply just recircularize, right? That's this top arrow here. We don't want that, right? So we use a phosphatase to cleave off the phosphate group, which will then prevent ligase from ligating it, right? So that way we have our plasmid stuck in that linearized form, and then we can use our DNA insert. This is our gene of interest that we want to insert inside of there. And now ligase can look for the phosphate and then allow those two to come together, right? And so then those nicks can be fused together again with other enzymes, right? So these are the sort of ideas you can think about, because you want to look at the chemistry and you want to look at, well, how do we solve those problems after knowing the chemistry, right? Awesome. Notice how you want to keep those phosphate groups on the DNA insert, right? You don't want the phosphatase to target those. So these might be an extra step, for example. Okay. So now, effectively, what have you done? I mean, we're not only becoming DNA ninjas, we're not only chopping the DNA, we're also making DNA. We're also fusing DNA together with ligase. So in essence, you have upgraded from a DNA ninja to a chef ninja, right? You're not only cutting them apart, you're also making them together, right? You're making new things. So congratulations. All right, let's move on. Here's a question. Can I join multiple fragments? So far I've seen, okay, I can just use one enzyme and I can cut it, ligate, now I have two fragments joined together, right? Plasmid plus insert. Can I join multiple fragments together? What do you guys think? Participants, yes and no. Or yes or no, you can't do both. The question is, can I join multiple DNA fragments together? Yes or no? I'm seeing 3 yeses. And Alex says yes too. Any brave souls want to say no? Yeah, you're all right. And so the answer is yes. I mean, I wouldn't ask a question if we couldn't, right? And so the idea is using something called Gibson Assembly. This is a very, very important procedure that a lot of people use because it's a very simple way. It's really just a one-pot mixture. You just throw it all into this master mix. And it all mixed together and you get this ligation. So how does this work? The Gibson assembly is a good way to basically fuse a whole bunch of fragments together. To be simplified, this diagram only shows 2, right? You have this DNA and you have this DNA, and you want to fuse them together. Notice how you want to design these fragments so that they have overlapping ends, right? You add these 2 to your test tube, and then someone really smart figured this out. If you add these enzymes in this way, you can get this action. You can first chew back on the 5' ends. This is where the exonuclease comes in, right? Exo means at the ends. So an exonuclease in this mix chews back those ends, right? Chews back this end, chews back that end. And so that means that this one's gone and that one's gone. And you're left with these fragments. Aha! Now you see the value of getting these 2 to be overlapping. Then they can now start hydrogen bonding, right? this orange part and this blue part can then anneal. That's the fancy word for come together, right? They anneal, and then you have some polymerase that will then start making new bases, and then your ligase then seals the nicks, right? So that's the process of Gibson assembly. So this is, as we saw, just fusing two fragments together, but it's very, very easy to get multiple fragments, right? If you see how at the end product you get these two fused together, we can simply just have a whole bunch of different fragments that all have overlapping ends. Let me show what I mean, right? Let's say we have these three fragments. We want to ligate together A, B, and some plasmid that we broke apart, right? It's very simple now, right? We simply just match up those ends. This one is complementary to that one, right? This one is complementary to that one. This one is complementary to that one. If you have all of these in the master mix and you have Gibson assembly do its thing, right? Exonuclease, annealing, polymerase, ligase, right? That just keeps on going inside of your reaction mixture. You will get all three of these circularizing together to form your vector, to form your final recombinant DNA. So this is a very good way to fuse together multiple fragments. So it's really, really efficient. It's really, really versatile, right? And so then you can transform it into your bacteria and have them become your cellular slaves, right? Okay. So any questions about Gibson assembly? You use some of the concepts we learned about in terms of, the regular restriction enzymes, but kind of building on top of it, right? Awesome. Let's move on. Okay. So here's the idea of genetic engineering. You can take your DNA, right? Think about how powerful this is. Think of any gene. Think of any gene in the entire world. You can put it in a plasmid, and you can put it in a cell of interest, and then you can have that cell express whatever DNA you want. It has all these different applications, right? It can be used in drugs, obviously, right? It can be used in anthropology, looking at really, really old DNA. It can be used in research. It can be used even in forensics, agriculture, right? All these different things are applications that come about from our understanding of genes and restriction enzymes, right? Look how far we've come. We've gone from looking at bacterial immune systems. Like, who the hell cares about that, right? We've looked at bacterial immune system, and we've gone through all these, fields. We've touched upon all these fields because of that singular discovery, right? Just goes to show how that basic science is really, really important. Okay. And here's just some of the many applications. Right? If you're thinking about doing research, here are some of the things that people have used genetic engineering to do in the past. So up here, top left, there's this thing called golden rice. And so if you think about more underdeveloped countries, right, there are a lot of people who are malnourished. They don't get the nutrients, they don't get the vitamins that they need. So they're very malnourished. They're not too healthy because of that, nutrient deficiency. So what scientists have done is they've gone ahead and they have genetically modified rice. And they formed this so-called golden rice. They have used these genetic engineering techniques to insert genes inside of rice to make them produce vitamin A, right? And so if, for example, a population somewhere has a vitamin A deficiency, just have them plant these, golden rice crops, and then they can, hopefully they can alleviate that symptom a little bit more. It's not just vitamin A, right? All kinds of things. You can also insert other nutrients. You can make plants and crops grow better in more extreme environments, especially with climate change. Temperatures are rising in a whole lot of places. You want to be able to have plants that are more hardy. You can have them by inserting genes that make them hardier against increasing temperatures. You can make them hardier against changes in the environment. You can make them hardier against salty environments, right? Make them halophiles. All these different things are applications of genetic engineering in terms of agriculture. Medicine, right? You take a human, you look at their gene sequence, and you find the gene that codes for antithrombin. Antithrombin is an anticoagulant protein. And so then you take that gene and you put it inside of a sheep, basically. So there are people who are antithrombin deficient. They have a deficiency in the so-called antithrombin 3 protein. And what you do is you take this antithrombin gene, you put it inside sheep, and the sheep is like, oh wow, I'm transgenic. And now this transgenic sheep can then start producing antithrombin in their milk. So all you have to do is take that sheep's milk and then isolate that antithrombin and then give that antithrombin as a therapy to these people who are antithrombin deficient. Very, very useful, right? This is really just the tip of the iceberg. I mean, I'm actually currently doing a project involving gene therapy, and it's involving these viruses, these so-called adeno-associated viruses. And there was recently, I think just in the past year or two, there were some gene therapies approved. And so there was this one gene therapy which involved taking a virus, and there are these blind people. They suffer from Leber's congenital amaurosis, and so they have these blindness symptoms, and so they can't see. It's really sad. And it turns out that their blindness is caused by the lack of a functional protein called RPE65. So then the scientists go, oh, well, why not just give them this RPE65, right? Let's put the RPE65 DNA inside of a virus. It's called adeno-associated virus, AAV. And they use this AAV with this RPE65 DNA. And they, for those of you who are not good with gory stuff, close your ears maybe, they inject it into their eyeball, right? And then you inject this, virus into their eyeball in the back of their eye, and the viruses go around in the back of their eyeball, infecting their retinal cells. And then by injecting that RPE65 DNA into those cells then start making the RPE65 protein, and those people are cured of blindness, right? It's a really amazing thing that can come from gene therapy. It might sound kind of gross, right? You're injecting things into eyeballs, but it's really, really cool, right? And so, of course, it's only one type of blindness. There's many, many types of blindness, but that's one application of this gene therapy, right? One more thing, criminology, right? Let's say you had a victim and you had a hair from the victim's shirt. There was some crime scene and you found a hair, and you use your restriction enzymes and you cut up the DNA sequences inside of that sample. And you run some, gel, for example. You run this SDS page, for example. And Colin went over these blots in his talk earlier on. We have a YouTube channel, check it out. And you run these blots, and you see how this is the victim, this is the hair from the victim's shirt, this is your sample, and you have two suspects. I mean, guys, who did it? One or two? Who is the criminal?
Two.
Yeah, obviously, right? It's very easily seen that suspect 2 was the bad guy because you can see how it matches up exactly with the sample from the shirt. You can use it for identification purposes, right? Because different people have different DNA sequences, but depending on the length of DNA between two restriction sites, you can get these different DNA profiles. And so DNA profiling, DNA barcoding is also really important. Okay, so all of these different applications with restriction enzymes. Now we get to Andrew's point, right? Now we get to Andrew's word CRISPR. And so I'm sure you've all maybe at least heard about CRISPR before in the news. And so CRISPR, it's very hyped up right now. It's the idea of using genetic engineering to very precisely and very easily modify DNA. And so I guess you can say it's a type of restriction enzyme. You can see how it's also described as these molecular scissors, right? Scientists really are just DNA ninjas going around the lab. And so you can edit genes, but that's not all, right? I'll tell you what CRISPR is in a bit. But first, we have to go all the way back to the first few slides, right? Let's think about bacteria, okay? And so before we think about bacteria, let's first think about ourselves. And so the reason why we're stuck at home, the reason why we can't see our friends, the reason why we can't go to schools is because of COVID-19. We have an immune system, but our immune system it can't fight against COVID-19. And we not only have our immune system, we also have two divisions of our immune system. We have the innate immune system, and we have the adaptive immune system. Let me describe them for you, okay? The innate immune system is the part of the immune system that doesn't really focus on identification. It just sees these patterns. It sees, oh, this thing is most likely belonging to the bacteria or this virus, and so I'm going to attack it. It's not very specific. right? They recognize things like PAMPs, so pathogen-associated molecular patterns, right? If it's just like a generalized molecule that some bacteria have, right? Maybe some kind of peptidoglycan fragment, for example, right? They can bind to those specific molecular patterns and recognize those as attackers. And so your innate immune system attacks those types of antigens. Your adaptive immune system is adaptable. It recognizes different nuances in your pathogens. So it can tell the difference between bacteria 1 and bacteria 2 depending on what surface receptors they have, what surface proteins they show off. So your adaptive immune system, that's your things like your lymphocytes, your B cells, your T cells, right? I won't go too much into that right now, but your adaptive immune system, the main idea is they can recognize specific proteins, and here's a key point, they have a memory, right? Your adaptive immune system can look at a pathogen once, and it will remember it. So if you get infected again, it will unleash all of its power because now it knows what it looks like. Now, restriction enzymes, the ones we talked about so far, would you equate them analogously to the innate or adaptive immune system of us humans? Text in the chat. Are the restriction enzymes we talked about so far innate or adaptive? So Alex says innate. Andrew also says innate, right? And that's correct. They're innate because they only recognize that specific sequence, right? For example, for EcoR1, or it was like GAATTC, right? They only recognize that pattern. It's almost like a pathogen-associated molecular pattern, right? It's a PAMP. So they recognize that specific pattern, and then they cleave it, and that's how they get rid of the virus, the bacterial phage DNA, right? So the restriction enzymes are innate. But what's even more fascinating is that bacteria also have an adaptive immune system. They have a memory, right? Just like our B cells and T cells form memory B cells and memory T cells, bacteria, they have a memory. And that's what CRISPR is. You hear about CRISPR all the time, right? You hear about CRISPR in terms of these, gene editing, feats, and they're going like, oh, well, I can edit this to this precision, right? It's really fascinating. But what it comes down to is the bacterial adaptive immune system. Here's how it works. Also, this is what CRISPR stands for. I don't know why they named it like this. Clustered, regularly interspaced, short, palindromic repeats. CRISPR, CRISPR. I don't know why. Like, why? Anyways, this is CRISPR, right? And so what they do is they look at a virus tries to infect it, and the bacteria is like, nah, I have these adaptive immune systems to fight against you. And so it uses those molecular scissors to cut up that virus DNA. And here's an even more really wacky thing they do, right? They take the virus DNA and they insert it into their own genome. Isn't that wild? They take the virus DNA and they're like, all right, I'm going to insert you into myself. Become one with me, right? They're very Taoist. Anyways, the bacteria now has this new spacer. We call it the spacer because you can see how this virus DNA is spaced from the other viral DNA sequences they store by this white sequence, right? So green, white, yellow, white, blue, white, red, white, pink, white, right? It's spaced. The virus DNA sequences are being spaced apart by these generic sequences. And so after this spacer is inserted, then it can be transcribed into this CRRNA, CRISPR RNA, right? And then it can associate with another protein. You'll never believe what it's called. A CRISPR-associated protein. Whoa, right? Whoever names these things, like, get on another level. Come on. Okay, anyways. So these CRISPR-associated proteins, or so-called Cas proteins, C-A-S, Cas proteins, they can bind with this CRISPR RNA. And then here's the cool part, right? If that virus ever rears its ugly head again and injects that bacteria and tries to infect it, now the CRISPR system recognizes it, it's complementary to it, and then your Cas protein can then cut up that DNA, right? So this is the idea of the adaptive immune system of bacteria, and we call it CRISPR, right? I feel like the public associates CRISPR with gene editing, and that's good, and that's true. But remember where it came from. It came from the basic biology of bacterial adaptive immune systems, right? Again, I ask the question, how can we use this, right? Forget everything you knew about CRISPR, and now looking at this diagram, right? Pretend you're a scientist who just discovered this. Pretend you're a synthetic biologist who knows that bacteria have this adaptive immune system that uses this CRISPR RNA to cut up specific sequences. What can you do? What can you use this for in terms of gene editing? Text in the chat or say it out loud, whatever you're comfortable with. I'll give you guys a hint, right? I'll guide your attention to the RNA portion of it, right? The RNA is what's locating that sequence of DNA to cut it up. How would you use that to your advantage? How do we
Can we
use
give
this?
it any mRNA we want so it can cut whatever we want?
Yeah, you got it. That's the really fascinating thing, right? It's programmable. You can give it any RNA you want. Well, I shouldn't say any, but like a reasonable sized RNA you want. And it's programmably able to find and seek that. It's almost like a spy, right? A spy chosen to seek out that particular target, that particular person, and spike them down, right? If you give this Cas protein an RNA sequence, it will seek out that particular sequence and cut the DNA at that site. Very good. Right? Andrew says we can just insert any sequence we want to cut into the plasmid. The bacteria will produce CRISPR proteins. Exactly. It's very easily programmable, right? Whereas for restriction enzymes, I guess you could mutate it and maybe hopefully get a different sequence, but for the most part, it's pretty fixed, right? Okay, that's the idea. Now, these scientists, I think they were a group at Berkeley. What they did was they looked at the structure of this Cas protein, and they saw how it worked. They were studying this adaptive immune system, and they saw this particular Cas9 protein. I mean, the most common thing you'll hear is, oh, CRISPR-Cas9, right? And so this Cas-associated protein, Cas9, is binding to this DNA fragment, and it uses, actually in the natural system, they use two pieces of RNA. They use what's called a tracer RNA, and they use a CRISPR RNA. So this is like the trans-activating RNA or something, and they actually use the CRISPR RNA and the tracer RNA together to guide this Cas9 protein to where it needs to be. And here's where it forms those hydrogen bonds, right? And then your CRISPR protein, it has these domains. I think it's like some domains like RUBC and AGM, and it cleaves the DNA using those domains. Question for you guys for understanding. Are these domains endo or exonucleases?
Endo.
Exactly, right? They're cutting inside the DNA strand. So these two endonuclease domains of the Cas9 protein are going to chop up that DNA, making a double strand break, right? I won't get too much into this part, but there's also there's a PAM sequence that has to be there as well. So it turns out that you can also kind of program the PAM sequence. It's a little bit more nuanced. But what these people did was, it's very simple. You guys could have thought of this, right? They simply fused these two together. They made an RNA strand that had a linker loop. They fused those two RNA strands together, and now you have a single RNA strand. They call it the SG RNA, single guide RNA, by simply fusing those two together. Ethan asked, is the tracer RNA not a linker loop? No, right? The linker loop is called that because it links these two RNA pieces together. In nature, if we hadn't stepped in, there are actually two RNA pieces, which if you think about, it's not too efficient for us because you have to make two of them, right? And then we have to worry about, oh, will those two find each other? Will they associate, right? It's very complex. And so by linking them together, we can artificially make this a more efficient system, and we can make it do our bidding. Hopefully that answers your question. Right. So this single guide RNA can then be inserted into the Cas9. Of course, there's some engineering that has to be done, right? Oh, will this loop impact the efficiency? And so after optimizing it, they allowed this CRISPR-Cas9 protein to do the same thing, right? It can go in and cut right there, and it works, right? That's the amazing thing. You can give it any sequence of RNA here, right? And it will go and search out that sequence in the genome of potentially billions of base pairs. And it will modify by cutting there and there, right? It's a very precise molecular katana or shuriken, whatever you want, right? And so, of course, off-target effects are a really important concern. If you are thinking about using CRISPR for clinical trials, you want to be careful about off-target effects, right? If it cuts somewhere you don't want it to cut, that's a huge problem. And so a lot of effort right now is going into, how to minimize these toxic side effects potentially. Okay. Now, here is another view of the Cas9 protein, Cas9 nuclease, right? And so you have your single guide RNA. It's one strand of RNA, remember, and it's fed into this Cas9 protein, and it hydrogen bonds to a particular DNA site, and it cuts at those places next to a palindromic adjacent motif, your PAM site, right? And so that's your Cas system. Now, again, I'll ask you guys the same question. I kind of gave it away because I showed you that next slide. But anyways, I'll ask you guys the same question. What can you do with Cas? What else can you do, right? Because right now it's just cutting. That might be useful, for example, for knockouts, or for example, to mutate it, right? That might be useful for that kind of thing. What else? Remember, the protein is your oyster, right? You can modify it. You can modify the Cas9 protein. What can you do? Andrew says insert genes with ligase. Yes, right? If you were to somehow able to, ligate a protein inside, and you were able to somehow cut it at one end to maybe form sticky ends, right? That could be a programmable way to make new restriction enzymes. What else? You can cut it. You can maybe ligate new genes, right? What else can you do? What's the new catchphrase? Cas9 is your oyster. Answer. Oh, can we have the cell make new genes, like combine new two separately cut pieces of DNA? I'm not sure about the ligation efficiency of the Cas9 nuclease, but yes, definitely if you want to cut pieces of DNA, that could be definitely a programmable way to cut pieces of DNA. Yeah, definitely. So here are all the different ways you can do, right? For example, as you guys said, you can potentially cut both sides. That's one way, right? We can also mutate one of the endonuclease domains to make a nick in the protein, right? And that might have some kind of other downstream consequences because the cell might respond differently for double-strand breaks versus single-strand breaks, right? DSB versus SSBs. You can interfere with that DNA strand. You can ligate, you can attach another transcription inhibiting protein, attach that to the Cas9, and disable all the endonuclease domains. So basically say, hey, don't cut, but find that sequence and bring my inhibitor to it. That will interfere with that DNA sequence and prevent that transcription. You can activate it, the opposite, right? What if you were able to fuse Cas9 and a transcription factor together, and you also disable the endonuclease domains? Then you can bring that transcription factor to where it needs to be, activating that particular gene, right? You have all these different things too. You can tag it, screen it, visualize it, purify it. You can do all kinds of things, right? Cas9 is your oyster. So that's the key thing, right? You can program in a whole bunch of ways. You can mutate here and there for the Cas9 protein. You can change the RNA strand to basically guide it anywhere you want into the genome. Think about it, right? Billions of base pairs. And your Cas9 protein, well, hopefully, will find one site, maybe one or two or three sites, within that billion base pair pool, right? It's like finding a needle in a haystack, right? It's really, really precise, really specific. Okay. Now, what have you done? You're not just a ninja. You're not just a DNA ninja. You're not just a ninja chef. Now, You are a DNA ninja who can morph his weapons, right? Now you can change your method of attack, right? Now you can change, oh, I want to cut right there. Oh, I want to cut one part. Oh, I want to cut these two parts. You can morph your weapon now, right? It's a really exciting time to be alive. But now, there are sort of more pessimistic side effects to this sort of hype, right? It is really exciting. You know, it's a really, really cool time to be alive in synthetic biology. But I'm sure you guys might have at least heard about this dude, right? He Jiankui. He was a Chinese scientist. He was a synthetic biologist who decided to mess with some embryos. And so he decided, you know, that he wanted to give these two children, I believe they were twins. Correct me if I'm wrong, but I believe they were twins. He decided to give them some HIV resistance. So he used CRISPR and he modified these embryos, right? The problem is that wasn't really necessary, right? There are other ways to prevent getting HIV. You know, there are some HIV cocktail drugs that are available, right? And so really this unnecessary gene editing, and also the fact that, you know, he did it without really any ethical guidelines. He didn't really get reviewed by an ethical board. He wasn't really, you know, scrutinized by any ethics whatsoever. You know, that was really led to his being sent to jail, right? So we're entering a new era of genomic editing, but at the same time, we have to consider the bioethics of the situation, right? Because this dude, right, I'm sure he did it with good intentions, but at the same time, you know, he did it to prevent them from getting HIV. But what are the side effects? Right? We don't know. And so you're really sacrificing, you know, these two fetuses lives, right, in order to do this experiment. So it's a really, really sort of rash decision to do. And so pretty much the entire scientific community was like, no, man, you can't do that. So pretty much everyone was against him. So now we get to bioethics, right? These last 2 minutes, I want to talk for a bit about bioethics, right? The idea is we have the tools, we have Cas9, we have restriction enzymes, we have ligation, we have all these different domains that we can mutate. We can mutate the endonuclease domains, we can insert CRAB domains for inhibition, we can insert transcription factors to activate genes. We could. Should we? Right? Last night I was watching Jurassic Park for the first time. And then in Jurassic Park, you guys might have seen it before. I know, I haven't seen it before yesterday. Really sad. But anyways, I finally saw it. And in Jurassic Park, what they did was they took the DNA from, you know, these mosquitoes. And so the mosquito apparently, you know, they bit the dinosaurs and they had the blood and they were, you know, preserved somehow. And they had the DNA from the blood and they made the dinosaurs back to life, right? And I think they also combined the DNA with the dino DNA with the frog DNA, right? And then that also had a whole lot of side effects. Now that's sci-fi, of course, right? But again, it got me thinking, should we? Right? Because if you think about it, we probably, there are efforts trying to bring woolly mammoths back to life, right? Those exist. There are efforts trying to do that. There are efforts trying to bring extinct animals back to life. The question is, should we? In the movies, that went kind of badly, right? A lot of people died. But yeah, this is just something to think about. Okay. So it's 8 o'clock. Are there any last minute questions? All right. I really appreciate you guys being here, you know, spending time out of your night and your summer to learn some synthetic biology. Really appreciate you guys being here. So if there are no questions, then I'll leave you guys to it. All right. I'll stop the recording.